Today's presentation is Juvenile Toxicology Isn't Just Mini General Toxicology, a conversation on how drug developers can navigate juvenile toxicology. Next slide, please. You can see that we have a disclaimer. I'll let everybody read that, but the opinions and views expressed in the following slides are those of our presenters and shouldn't be attributed to outside groups. Thank you. Next slide, please. I'd like to welcome our speakers for today, Dr. Susan Laffin and Dr. Christina Reardon. Christina is currently a Senior Director of Integrated Drug Development at Certara. She has over 19 years of experience serving as the toxicology and safety lead on multidisciplinary drug development teams. She's worked in many therapeutic areas, most recently with a focus on immuno-oncology and inflammation. Susan is currently the head of non-clinical global investigative safety at GSK. She has over 18 years of pharmaceutical industry experience and is an expert in reproductive and developmental toxicology. Christina and Susan, welcome to today's presentation. I'll now turn it over to Christina to begin the presentation.
Thanks, Suzanne. And hi, Susan. So glad you could join us and share your experience with juvenile tox studies. I get questions about this all the time. So we have a lot to cover. I'm just going to jump right in, but we're going to start with a couple of audience poll questions. So the first poll question, there we go. Who has supported a pediatric drug development program? We'll wait a few minutes so we can get the tally. Oh, there we are. So 66% of you have supported a pediatric drug development program. I think that's a little bit more than I was expecting. And our second question is going to be, who has run a juvenile tox study in any species? And these polls are just going to give us an idea of the breadth of experience of our audience members. Oh, okay, so that's interesting. Haven't run juvenile tox study, but had more people actually support a pediatric clinical trial. So on that note, we're just going to jump right into the rest of the presentation. And I think the first question that everyone has is, how do I know if I need to run a juvenile tox study? So Susan?
Thanks, Christina. And I just want to say thanks for inviting me to come today and talk to you about, one of my favorite topics, juvenile toxicology. And I'm really inspired by the poll answers I just got, which is fantastic, right? So 2/3 have already said that they supported a pediatric program, but then only 1/3 had actually run a study, which gets right to this question, which you should be starting your whole discussion on, do I even need a study? It is not a default assumption that you need a study. Right, and so the first place to go to look for that answer about do I need a study and where to even start, I would recommend you go to the ICH S11 guidance that's been recently finalized. And it starts with the main objective of answering that question of whether or not you need a study and what you would do. And the answer to that is it's a case-by-case bespoke decision that needs to be made, because pediatric investigative plans are so variety and so much difference in their approaches and what the situation is, especially with respect to risk benefit, right? And then the other half of this guidance is really like, if you did need a study, we'll give you some guidance on what you need to design that study, and hope that'll be the topics for today. So just also to mention briefly, we can talk about it more later, but what's in scope for the guidance is, monoclonal antibodies, biologics, small molecules generally, except for those that are in supportive oncology that would fall under S11 or S9. So if you fall under S9, you go to that decision, that guidance. In their Q&A, there's some answer there, a little bit about whether or not you need a study, and then you'd come back to S11 if you were to do one. Also of note, the cellular therapies, gene and cell therapies, and vaccines are out of scope for that. In that guidance, one of the main figures that I'll just spend a little bit of time orienting to is this weight of evidence approach to decide whether or not you need a study in the first place, right? And so just to orient you here, so there's these weight of evidence factors in the blue circles, with the darker blue ones here being your two primary ones, and they are actually weighted. These two are weighted heavier than the four below it. The four below are in no specific order, but these clearly are heavier weighted deciding factors, right? And that is the youngest intended pediatric age, right? So that clearly is the first component of how young the youngest age would be when you're finally done developing. How young do you have to go, right? And the younger you have to go, the more likely you'd need a study, right? That's kind of how these bars work. And then you just go through each of these factors, right? And then, you know, is there effects on developing organ systems? Yes or no, or there's a big uncertainty. If that's a scenario, you'd be more likely. And you can kind of see how you go through that pattern and work your way from each scenario, which I do have a few to share real quickly today. So this would be a case study where you were, you know, only going into adolescence. You don't have any known effects of developing organ systems. So you're already starting out here, right? Less likely. So with these two weighted factors, it's looking like you would not propose a study would add any additional value to that decision, right? But you can go through these as well, just to have you oriented. So at least the pharmacological target has a role in organ development. We're saying no, it does not, right? So that's more a theoretical exercise you can go and do if you haven't done yet for the adult indication. The modality, meaning, is it, how are you altering that target, you know, effectiveness? So is it a monoclonal antibody, which would be like a highly selective approach, or is it a small molecule that would be like a lower selectivity where you might have up target? So that using those types of things, you would say, okay, even if it is a small molecule, you'd be over here, it's a low selectivity type of modality. And then a 1 month clinical duration that's kind of in the middle, and you do have adult clinical safety data, so you're over here. So here you'd have kind of the weight of evidence is telling you that you would not conduct a juvenile toxicology study. And clearly some of you have already been through that, since many of you said you supported it, but you haven't run a study, or maybe you're still waiting to do it, but that seems like people are understanding that now, which is great. And then the other case, again, a little more clear cut. So here you're going into a younger child, 1 year of age, right? So now you're over, it's pretty far left on here that the more likely to need a study, but you don't have any specific concerns, so less likely. So then you kind of have to go a little bit further, right? So is there any other concerns, theoretical or otherwise, for the role in organ development, knowing what you know about the pharmacologic target? And so yes, there is. There's a possibility there. You know, we're going to use a small molecule. There's another reason. The treatment would be chronic. There's another reason. You know, so now it's coming into, you know, more like you would propose that a study could add value to that risk-benefit discussion. And usually in most cases you have some, you know, limited data. You've gotten to maybe a first time in human, or you have some clinical safety data, but not a lot yet. Okay.
All right, thanks, Susan. Those are great examples. I always prefer to see examples to help you put things into perspective. So now that we've already figured out that we need a study, how do we know when is the right time to run this study in relation to your development plan? And is there anything we need to consider specifically to support that clinical plan?
Yeah, thanks, Christina. I'm going to actually turn off my camera now. I realize I hadn't done that, but we'll see.
Oops. I think you muted, Susan, instead of your webcam. Wrong button.
Great, thank you. Now we're good. Can you hear me and not see me?
Yep, you're good.
Okay, great, thanks. All right, so the question was like, when would you do a study? And I want to point to, you know, the answer is always in the non-clinical spaces. You have to understand what the clinical program is doing with the clinical spaces, you know, what's their plan, and then you make your non-clinical against it. And so that you need to kind of keep having those discussions at the team level and with the pediatric specialists. And one of the guidance they should be very aware of is E11, just happens to be 11 as well, but that's the clinical ICH guidance. And in there, they do a really nice job of categorizing the age groups for you. So you can, have these discussions down to the specific age groups and know, when clinically you will go through those groups. And then from there, we'll use that to decide when you would do your non-clinical studies to support them. Right? So there's a good framework you need to work against. And it's definitely, this guidance is really good too, just to give you a broader scope of the things you need to consider that are different in pediatric medicine, right? So the ethical issues are different, the formulations are typically different, right? And the types of studies and the considerations are just very different in here. Clearly, you're negotiating with the parent, right? You have to, the consenting is different. It's really assenting, right? Whether or not they would put their child in a clinical trial. Oh, I think I went too many slides. Okay, and just for that, I want to just spend a small time on waivers and deferrals. Those are terms used towards the pediatric plan, right? Not towards the juvenile toxicology studies, right? Because you get a waiver against these because the guidance or the regs, depending on the region, require that you address each of those age groups that I just shared with you about. So then if it's required to address it, you need to specifically get a waiver in either all the pediatric ages or a portion, right? So you can get a partial waiver if it's just 2 years old and younger, and then go through some of the reasons. Sometimes there's just no clinical need because the disease indication is not appropriate, not classified towards children. So you could get a class waiver, and then also there's product-specific waivers, right? And there's 3 different grounds that it's only occurs in adults. So the children or children of certain ages don't get this type of disease, so they wouldn't be applicable. It can also be that it's considered potentially unsafe, that there could be a safety related waiver. And then also there could just be, depending on the region, it might not actually be something above what already is happening for standard of care, right? So that's the decision about a waiver. So that's not to address that age group at all for those reasons. And then there's deferrals, and that's deferring the age groups to aligned against the adult file. So this is in kind of a typical situation where you have an adult indication and then you're coming back and developing for peds. So there has definitely been a push over the last 10 to 15 years to get medicines to children, sooner and safer, right? So we're kind of earlier in the pushing the pediatric development earlier, and it's not always just a phase 4 type of thing. So you have to go through and have discussions about deferrals to have it after registration. So you can get partial deferrals, or full deferrals in, the whole plan, but that needs to be fully understood before you can put a proper timing against when you would do a non-clinical study or juvenile toxicology study. And real quick, and I'm definitely in the kind of saying that there's a typical timing when they're, I'm saying these are also very bespoke, but just to get a handle and just to say some of the things we could commonly see. So you have the drug chevrons here, very basic, phase 1, 2, 3, right? With the application file happening, after the end of phase 3. And so the plan might be something where you certainly want to seek advice early, around phase 1, phase 2, get your investigative plan going, and then likely consider including adolescents in phase 3 without doing any further non-clinical work. But then post-registration, you'd be going into the younger populations in some deferred way, right? So here I have 2 to 12-year-olds, and then going younger than 2, like 6 to 2-year-olds. You do some sort of staggered deferred plan. And before you'd initiate studies, let's say this, like under 12s, then you would be conducting your juvenile toxicology studies and dose ranging that study. So you would be, just as note, these studies are typically done much later in development than other toxicology studies. So there's definitely some different concerns for that. If you can think sometimes even during phase 3 or after registration, you have lots of sometimes global open large phase 3 studies going on, and you're producing new toxicology data. Okay.
Okay, great. So you've already touched on a lot of the regulatory requirements, but I want to move into the next audience poll. Who in our audience has written a pediatric investigational plan or a PIP or PSP? Okay, everyone done voting? So So 36% of our audience has written a regulatory plan. So on that note, Susan, you want to launch into our regulatory requirements?
Yeah, I will, definitely. That's great. It's interesting to see how many people have experience with them because the PIPs, let's see if I can move to the slides and show. We're getting there. Hold on. Okay. Yeah, so the perspective, we put the PIPs and PSPs to see that you recognize those acronyms, right? And that the PIP is EMA, a European-based procedure, and the PSP is the FDA-based procedure. But really, from a pharmaceuticals perspective, the sponsor, we're trying to get one plan that's globally aligned, right? What's the most efficient? What's the best way to develop the medicine and not be cumbersome about it and try to get, like I said, medicines to children sooner and safer, right? And so sometimes it is quite a process to go through these and get alignment. And I'll just say to try as much as possible to keep those going as earlier and develop as you can. And you can have, there's more and more discussions happening between FDA and EMA, and you can have them attend meetings. I know they have some. You can also ask them to attend certain meetings together, so you can kind of have a more unified discussion. And one last bit I can offer is you can also submit some of the meetings, or if you have an agreed PIP, you can submit that in, to the FDA, or sometimes even other health authorities who aren't part of those larger procedures will ask for those, and it's just a better way to try to get a global plan and be efficient about it, right? So I encourage you as much as possible to do those. And I'm not going to go into the specifics on the regulatory, all the legislation that's available, but clearly there's a large legislation body with the EU and the US having very specific binding agreements. So this is something very different than in general toxicology. You will go through and you'll have these PIPs, investigative plans, have non-clinical and clinical binding agreements, right? And so they will be checked and compliance against. So those are very important. You need to put all your experts in and have that discussions earlier there and not agree to things that you're not fully prepared to do. The other guidance is I mentioned already. So there's ICH, E and 11 to remember, E and S 11. There's lots of good information in there. And then just to mention also the World Health Organization does have some initiatives about make medicines child size and trying to get globally more medicines to pediatric patients. Again, I was just going to go back to our timeline. I've already been mentioning, to try to engage the health authorities earlier, the better. That's certainly a plan when you have some discussions with them. Sorry, train going by. Can't mute that. Okay, so then you have, again, your chevrons of drug development. And I have up here on the top is FDA and down below is EMA. So FDA has the pediatric study plan, right? And so there you can go get early advice and you can have some dialogue, maybe type C meetings for the adults. You can ask a few questions in there. But they have a pretty hard line in the sand. That's a legal requirement. That's 60 days after your end of phase 2 meeting for the adult indication, you need to have your PSP aligned and submitted, right? So they have a very clear, crisp requirement there. And then you're going to see there'll be checks, and there's also different, I'm going to go through the specifics of it, but this is best Products for Children's Act, I think, right? And then there's also, you can get written requests. So there are some specific things that can happen there as far as getting the pediatric plans going and compliance checking against them for FDA, right? And then for EMA, the quest is to do it a bit earlier and have some more agreement earlier, but then it's not quite such a hard line. And I think sometimes now it's going more to the end of phase 1 or right into phase 2. Like once you have reason to believe you're going to be able to develop this medicine and some good information about it, you can get your PIP agreed and submitted, right? And then there will be compliance checks against it at the time of registration or otherwise, right? And these things, I mean, it's fully recognized that they're going to change as you go, right? So there's definitely amendment procedures in place and things that you can do because as you go through this, it's a dynamic process.
Okay, thanks for all that really good information. That's always a question that comes up is when do you file these regulatory plans? But now we're going to switch gears a little bit and get into the specifics of the studies themselves. And we're going to start off with one more poll for the audience. And this is for those of you who have run juvenile tox studies, what species did you use? And you can choose more than one here. So we'll give that a little bit of time for everyone to put in their choices. Okay, there we go. The results are, so the majority of people have used the rat, and then let's see, we followed by monkey. That's actually a surprise to me. And then dog, rabbit, and other. I'm curious about the other. And on that note, we'll just roll right into some species difference.
Thanks, Christina. Yeah, that's really interesting, but it is mostly aligned with what we know from the other database searches, or the rodent is the most popular. Okay. Yeah, so also when you start to select species, I thought I'd just have this more as reference for you to use if you want. It's really just a kind of informal collection of information that we know from working with the contract labs. The CROs are really the experts on the delivery of the study type. But you can see in the rodent, you know, how early what's feasible to dose these animals, you know, so by gavage or intramuscular sub-Q, those are what we're saying here. So, and, you know, you can do it as early as the first day of life, right? At least for these ones, and then a little bit older to do infusion, either bolus, you know, or IV infusion. And then we have it here too for the dog. It's possible, you know, gavaging them maybe a little bit older, but then you can do intramuscular IV younger. And we didn't see many mini pig studies, but I have the mini pig in here as option. Right? So one of the things is just because it's feasible doesn't mean it's recommended that you do it, but you just can understand that there are a lot of feasibility questions to understand there. And here I say you can do it in rabbits and monkeys, but they're considered rare. And I think the poll today certainly showed rabbits, you know, not as frequent. I think it was like less than 10% or so, but there was a good bit on monkey. And I'm wondering if those are monkeys that are kind of adolescent monkeys, really, because if you think about it, you know, we rarely wait for mature monkeys, or if it's really doing a dedicated juvenile NHP study. So it'd be interesting to get that feedback. I do want to say, just for like typical, I use these words, I say typical study designs, but then right away I say there's no default design. So I get that I'm kind of talking out of both sides of my mouth on this, but just to try to get some basics, you know, that I've seen in the experience that we've had, you know, and certainly that the database has collated these so far, is like one species is generally sufficient. The rat is by far the most common of those one species where juvenile toxicology studies are conducted. And when you're doing your species choice, definitely make that aligned. If you have existing toxicology data, it certainly would be good to stay in those species so then you can compare adult versus juvenile animals. It can be a very informative comparison. You know, and then the age that you start the animals, it's really very much pinned to the human to animal comparison on development, right? So it's really, what is the youngest patient age that you're to support, like I mentioned before, clinically, right? And then go from that age in the patient to what would be, you know, a reasonable comparison to the species that you're going to work in, right? So overemphasizing that I'm taking the leap of kind of generalizing here, but in the rat, typically postnatal day 7 or 9 seem to be common start times, and in the dog at 3 weeks of age, like post-weaning, right? And a lot of times it's because, as I mentioned, the weight of evidence, if you're going to the younger patient population, you're more likely to need a study, to run one. So then you're more likely to be supporting, you know, a 1-year-old or 6-month-old clinical pediatric patient, right? So then you're getting into this age group in the rat, things like that, right? And certainly, I can't say enough, you need TK assessment should be included.
Oops.
Why don't you spend a minute on this figure, I think, first here?
Well, I wanted to ask you a couple of questions first before we jump into that.
Okay, great.
Yeah, it is a two-part question, and you already sort of touched on it, so I'm glad you're going to move right into that. So we tend to start our general tox studies, especially in rodents, with pretty young animals because they develop and age more rapidly. So my first question is, what ages are covered in your general tox studies without having to do a juvenile tox? And also, with the more rapid development, how do you design dosing in, say, a rodent study or any of the species to support your clinical plan?
Okay, yeah, great. Thanks, Christina. Right. So those are really good questions. And, you know, they're ones we see a lot, right? So I'll address the first one about study overlap. And maybe I don't need to go to the slide straight away, but it's very common, like I mentioned already, certain non-human, in the non-rodent species, in the dog and the primate in particular, and you've maybe conducted those studies already. And you're like, well, we started in relatively young animals. You don't always wait for maturity, certainly a non-human primate, right? There's actually a big demand and it takes a long way to get to mature monkeys. So So oftentimes those studies are actually conducted in adolescent animals. And I think, if you're going to do a clinical program in adolescents, especially if you have a deferred strategy, it's you could really take a hard look at that information, and you likely have good support already because you've exposed and assessed the toxicity profile in animals at appropriate age to adolescents and above. I have this was the figure I was jumping to before earlier, but I think although it's a busy figure and I'll spend a little time, it does really give you the a flavor for what decisions you need to make, right? And so this is like over comparing the rat. So the rat is kind of on the top half, and the timing of development of the different major organ systems, and then the human down below with this timeline across here, and then kind of these buckets that I'd mentioned that E11 describes, and the ages. So these are the clinical ages from neonate to infant, child, and then up to adolescent. And then the way the blue shading is working, when there's a darker blue shading, that's like when you have, very distinct morphological development happening, right? So very organ development is actively happening there. And then as it shades off, it's not perfect, but it's just, we did spend some time trying to get this close, where it goes into the lighter blue, which is more like functional development and kind of the fine tuning of the organ as it's developed, and then off into the pair into the white until it's near complete, right? So you can see when you pick an age across animal to human, you're never going to, it's not going to be perfect alignment across these organ systems. So you need to make trade-offs about which ones are of more concerns or which ones you want to. So you're doing that at both times when you're deciding when to start initiating and how long to dose the animals. If you had a certain organ system you were concerned about, you need to dose over that window, right? And then again, when you get your study done and you're going to interpret it, you probably should go back to something like this or get back to that mindset to understand the relevance. And then your second question, I thought I want to first start off. So that's about when we decide when we're going to dose them, or that we need a study and you're going to dose them at an age appropriate time. And I want to very quickly say that when you just make these decisions about dose duration, it really does depart from kind of everything you're taught and trained as a general toxicologist in the adult space. There's a chart there in M3R2 where it says, like, if you wanted to dose for 1 month in the clinic, you have to dose for 1 month in your non-clinical studies, right? Or you want to do chronic studies, you got 6 months. And so it's very 1 for 1, and it's very clearly described. That is not going to be helpful in the juvenile toxicology, because it's about, as I mentioned, exposing those windows, those critical windows of development, right? And when you have active growth periods, right? So it might be that you need to dose, you might only have a 10-day or a 2-week clinical dosing paradigm, but you want to be able to treat children from 2 years old up to adolescence. A 2-week dosing paradigm in the rat, even though they grow fast, is not going to cover that span of development. And I always propose you should try to do one really good study instead of a series of other ones, and try to cobble them together. So I'd rather dose longer from the rodents, from something comparable from a 2-year-old to an adolescent, and then use that information to support all of the clinical program. I have a note in here, I already spoke to it a little bit, about don't dose younger than you need to, because it gets very complicated very quickly the younger you go, just to get your doses right, and tolerability issues and technical issues, like really, so very, very generally, again, I'm just putting this out for, a little bit of help on the guidance on it. But if you're going to do a 2-year-old, you can do post-weaning in the rodent, about a 1-month-old infant, like I mentioned before, postnatal 7. And we have experience, starting on PND4, and that's supporting all the way down to a preterm infant. So there's really no reason to go younger than PND4 in the rat, unless you're going to go, we've done it once, considering like a gestation, like if you're treating a pregnant woman, right? And I've already mentioned it here a little bit. The age when you finish dosing, you want to make sure you capture that maturity window, the critical window of maturity there. And also, if you want to be assessing, sexual organs, you need them to be mature before you can assess them properly. And so the one that kind of hangs there is that looking at the histology. So you need the males to mature enough, like to get to postnatal day 60-ish or so, before you can get good testicular histology on them.
All right, great, thanks. Again, I'm really happy that you give some good examples for us. So now that we're into our study, we got it started running. So let's talk a little bit about our study endpoints. What should we need to consider and what we're putting in for our study endpoints?
Yeah, so for when you're running your study, I think when you write in the objectives, the two that really stand out different that are unique for juvenile toxicology is that there should be an objective that you're going to assess that any potential effects on growth. This would be the one and only study you really would be targeting to look at growth as an endpoint. And then again is development. And so depending on the duration and the species and how long you're treating, right? But any development that's key that happens during that time window, you should consider assessing. Most commonly, this ends up being, assessing the timing of puberty or hallmarks, physical hallmarks that are, landmarks of puberty, but you'd put those in there. And then, of course, it does kind of come out like a general, this is where it's a general toxicology study where you're doing those main assessments and you're assessing the toxicity, right? There, you're specifically looking for any new organs or different types of toxicities, or even just really distinctly increased sensitivities from adult animals. So those are the main objectives, right? And so that's what you need to understand, your objectives, and then that will dictate, the endpoints you put on the study. Here I'm going to go back to looking at ICHS 11, where there's the whole second half of that guidance is really helping the design of the studies, and they've come up with like a different approach where there's core endpoints. So it's expected that your definitive juvenile toxicology studies would have these core endpoints in most cases, right? Otherwise you've agreed something different. And then you have additional endpoints. And so those additional ones are only added on by having a specific safety concern, and there should be rationale to add them. So that kitchen sink approach of putting everything on because one health authority might want it or one might not is really discouraged. These studies can get very, very big, and you can kind of compromise being able to just deliver them and and interpret them. You don't really want to put everything on there and kind of make it fall down on its own weight, so to speak. So the core endpoints would be your classic general tox type endpoints that you would do in life, and then at the end of the study, right? So you'd have ClinPath and some your main study necropsy, right, where you'd have your macroscopic, your organ weights, and histopath, right? So that would be part expected. TK, certainly a key important piece of information. The assessment of growth, like I mentioned, you would put on a long bone measurement. There was good alignment already. That was one place where the health authorities had alignment to measure the long bones as a measure of growth. That needs to be interpreted with body weight, of course. And then you'd put on, like I mentioned, any developmental marks you could, physical landmarks of sexual maturity, right? And these ones, you can noticeably, all of these kind of major ones that add more animals, typically more complexity to the study, you would do by identified concern if you're going to add mating on them or other fertility assessments. In S11, I want to mention for females especially, you can just add potentially looking at their estrous cycles rather than a full mating. You can just make sure they made it to maturity and that they're cycling normally. That would be one way to address it too, without doubling the number of studies to make them. There's a good section dedicated to CNS, right? And you can add immunotox endpoints. Recovery or off dose, that is one I think there was a lot of debate, quite frankly, during S11 on that. And it is listed as an additional endpoint, but I think the expectations there are a little bit higher. That you'd more strongly consider adding in a recovery or off dose animals. And then, of course, you can do anything that you need to if you have specific concerns and you can figure out how to address them on the study. You're encouraged to do so.
So now we're on to the question about the litters with endpoints. That I know it gets really complicated managing litters, and I know that there's a lot of really important considerations that people might not think about, especially if they're more used to running a general tox study. So let's talk a little bit about how do you handle the litters?
Yeah, thanks, Christina. And I think this one I do want to spend some time on, and it is certainly something very unique to juvenile toxicology studies. And it's a bit of a logistics puzzle, but it's really critical to think about what you're doing when you're making these decisions about how you set up these studies. And I know the CROs typically deliver most of these types of studies, and a lot of people can defer to the CROs and certainly need to work with them, but you need to understand what they're doing because there isn't agreement across industry. I know I've worked with the major CROs that I'm aware of. They have different approaches to this, and there's pros and cons to them, right? And so there's not a unified agreement on that. S11 ended with saying, here are the confounding factors, please have a good think about them, and did propose a certain approach, but it's not mandated, right? You can do other approaches as long as you're understanding what you're doing in those and their effects on the confounding factors, right? So just to orient you a little bit to this is the kind of the three major decisions you need to make, like when you're going to bring animals in and figure out how to deliver the study, right? So the first one is when they litter out and you have the litters and the dams. So this is all talking about typically rodents. you decide either to keep the litters in their natural state, so that's to keep the moms with their offspring directly, or the fostered approach where they mix up all of the fostered pups and bring them back to the different dams, like with the intent, it's arbitrarily mixed up, because now you're talking about many animals. Logistically, it's a lot to do, right? But then now you've kind of broken that genetic link, so they're not with their dams. And then once you have the litters and you've got them to the size you want them and the age you want them, then you have to decide, are you going to dose one litter all the same dose level, or are you going to dose within the litter control, medium, and high dose level, for instance? And there that we have a little bit stronger guidance that dosing the whole litter the same is the preferred way. So you don't have to have so much cross, you don't have control animals and high dose animals in the same litter. So like if the high dose animals are, having some toxicity and not faring as well, they're kind of competing with their control litter mates for suckling time, and that's probably not ideal, right? You're going to kind of overly confound that situation. But I know some labs can do it. And then here, to your real question was like, how do I assess endpoints, right? So then you can do the endpoints like I said interlitter, so that's doing like taking one per each litter assigned to an endpoint, or taking the whole litter and putting that whole litter towards one endpoint. So those are all kinds of decisions that need to happen, and you need to know what's happening in each of these groups, and it should be in the protocol or the study plan, and it should be clearly described in the report so we can see what was actually done. So the study could be recapitulated or understood exactly what was done, and then you can know how they were, what you were controlling for, and you need to know all of those pieces to know what you controlled for. So some examples. So this is the option that I prefer, and it's one where you do have 10. So you have, these are large. Let me get you oriented before I go into too much. Right. So this would represent one dose group, and each of these hash marks is one pup that is part of this litter. So in here you have 5 males and 5 females in one litter. Right. And then there's 10 litters here, and then you would keep them in their natural state. So now you know who their connections, their genetic connections are. Dose the whole litter the same dose level, and then assign them the endpoints intra-litter. So like you would take one male and one female from each litter, right, and put them to the main study necropsy, right? And you can do the same with TK, right? And then the black ones are the recovery animals. Right, so when you do that, you've controlled for genetics because you're taking the one male and one female from each litter, and they're genetically linked because you have not fostered and mixed them all up. Right, and you've controlled for maternal care because you've taken only one from each. So you've used one out of 10, you have 10 different moms, so to speak, maternal care, right? You spread across for that. And you're keeping your litter size and your sex and your treatment similar across the groups, right? Across each of the litters. Another approach that I've seen used is where you foster all the pups. So you mix them all up. So now you do have the genetic, you've controlled for that. You treat the whole litter the same, but then you assign the endpoints to the whole litter, right? So logistically, that's a little bit easier because now you've taken the whole litters all come off right at the same time. But many times these are brought into adulthood anyway, right? They're separate, but you would just take all of these to main study necropsy and use these litter numbers for TK and these litter numbers for recovery, right? So that addresses genetics, and you're careful about it. You keep your litter size and your sex and treatment controlled for it, but it does not address maternal care. At least it certainly doesn't advantage of all of, you've brought 10 litters into this dose group, but you're really only using like 3 of them to take to main study necropsy. So you've kind of concentrated the maternal care component. So if one of these 3 is not as great of a maternal mom, then you can see where you could get litter comparisons, litter confounding effects. Whoops, I hit the button too much. Okay.
That's great. So obviously this next topic here is about dose range finding, because I do get the question sometimes, do I really need to run a dose range finder study when I already very well have a well-established dose response in my adult animals? So what is it going to add to do a dose range finder?
It adds a lot, and I can say yes, you need a dose range finder, certainly if you're going into pre-weaning, right? And that's strongly recommended that you do it. And I get that there's a question, do I really need it? Because you've really gone through and you understand the profile in adult animals, and the answer from our experience is yes, you do. Their surprises are more often what happens. And I can say, first of all, it can be challenging in these juvenile toxicology studies to get your dose selections correct. And the main goals, right, is to have, which is standard for adult study, but it's just in the same goals that apply here. They're just harder, I think, to execute. And that is keeping the exposure separate from your low, medium, and high dose, right? You always want to do that so you can interpret your data well enough. And then keeping it consistent throughout the duration of the study. I mean, sometimes you get like accumulation and things like that, right, where it's difficult in adult animals. But here, it's expected almost that the ADME differences in the animals is going to affect the exposure throughout the duration of that study. So you might have to consider dose adjusting or making sure you understand the TK in there. And dose ranging studies certainly help you navigate that before you would dive into a large juvenile toxicology study. Some other factors for dose selection. The steepness of the dose response curve that you already know about and the adverse effects, you certainly want to be considering that when you're going into younger animals. Pay attention to the margin you can attain or get over for the predicted clinical exposure, right? If you do have the pre and postnatal study already conducted or are conducting it, take a close look at that and pay attention to any findings and things you might have seen in that early postnatal part of the study, right? And then I just want to make a note too here about the high dose and your dose selection. It should be lower than what you would have in adult animals, right? Because these are growing animals, they're gaining body weight. Body weight loss in a juvenile toxicology study is way too much toxicity, right? Because if they're losing weight, then you're looking at them being, there's more abundant mortality happening pretty quickly to that. You just want to have like a slight decrease in that gain, and they're, gaining weight rapidly. So I would be down in here, like a 10% decrease in body weight gain would be really, a good place to target. And if you got a little bit less, that's okay too, because you haven't confounded all these other components you're going to look at in your study. We know if you affect body weight gain too much, you're going to have confounding effects. One more note too is that when you do your dose ranging findings, please have agreement on what your youngest age of intended population is, so you know how young you're going to start dosing those animals. You can dose range, but if you've not dose ranged in the right age group, you could be put, kind of falsely setting yourself up. And some just things to share, like here, little examples. So let's say this is the adult data. Here's the dose levels, kind of typical dose levels. This might have been like 25, 75, 100, and 300, right? And the adult males, you don't get as increase in exposure, but here there happens to be a sex difference in exposure. It's not too uncommon in rodents, right? And the adult females are just, they're getting much higher exposure as you go into the high doses. And then the question is, okay, well, now I'm going to go into juvenile animals. Are they going to be more like females? Or are they going to be more like males? You don't know, and you can try to predict it, but here's an example of this. We're not like neither really, right? So you get into these very young doses, very high exposures, and this is on postnatal day 13. The males and the females were similar. It's very rare. I haven't seen it yet where you have a sex difference in exposure in a pre-weaning animal, right? Because they really haven't done the sexual differentiation. So there wasn't a difference between males and females, but certainly there was a difference where they're getting much higher exposure for a much lower nominal dose. Right? So if you didn't dose range that, you would have some pretty bad surprises in that main study. Here's another example. I just have them over the top. But if you have no difference in sexes right between, so the adults, and you have this really nice adult linear exposure happening, and then in orange is the juvenile animals, and here, like postnatal day 4 and 13, it's very low, like it's just not getting on board, right? So here you could run your dose range study and think everything, they're tolerating it really, really well, but then as they age up, they start to be more like adults, and now their ADME probably similar to adults, something like that, right? Here, this ended up being an absorption issue. We think it was crashing out in the pH difference, as a pH dependent solubility, and we were just not getting it on board. But if you had not done TK in that early study, you wouldn't know that, right? So you need to get TK and you need to dose range. Those are some examples for that. And then this is what we've over the time come to, is like not only dose range, but really do a small tolerability study to try to avoid these surprises and putting a lot of animals on it first. So the main theory of this is start small, and get some tolerability information, and then go into a larger dose range study. And we don't do any controls, and just 2 litters per group, just so you're not only doing 1, right? And then just dose for a shorter period of time, get some TK and get some tolerability. And then from there, that can kind of inform your larger strategy, right? So if they were kind of somewhat more sensitive, but you think you can do it, then maybe just do a little bit larger dose range study, kind of more what people expect for dose ranging, and then go into your definitive. No surprises at all. You can go straight to the definitive. I haven't been lucky enough to run that route yet. And then you can get this where they're markedly different, where you really know you have something to navigate. And so when that's happening, you either need to figure out you're going to dose adjust or you're going to split the cohorts. Just remember, if you split the cohorts, you're now like doubling the number of animals you need to deliver that. But certainly you're going to have to do more dose ranging if you're having some surprises where you can't tolerate the animals can't tolerate it, and you need to understand a little bit why, because that could be giving you a cue that's important to the peds. And here's just an example. So this would be like a dose range study, right? So this is a low, medium, and high dose, right? And you can see as the animals age out, PND 13 to 22 to older, like they're starting to either metabolize it better or eliminate it better, you know, and the doses, the exposure is all going down, right? And so you can't really have that happen on your definitive study. So you would do something here, you kind of adjust, right, post-weaning or right near post-weaning, and you can, I kept this, it's kind of messy, but that's how it is. Like, you're not going to overly fine tune this to make it perfect. Like, one good adjustment to try to keep like your medium dose group, you know, so yeah, they start at 10 and it's going down, and then you're jumping up to 20 and then it's still going down, but you've kept them above a range that you're keeping your low dose group, right? The dotted lines is where they would have gone had you not adjusted. And the same thing with green, right? It's going down, jump up, right? So it's trying to explain that it's not going to be perfect, but it will at least keep them closer and separated.
Thanks again for those examples, Susan. I don't always think about a tolerability study prior to a dose range finder. But now that we've run our perfect study, let's get into data interpretation. What do we need to think about for data interpretation that might be different than what you would think about for a general tox study?
Yeah, so back to like, right, interpreting, you're going to go through your main objectives, right? These are the ones you're going to look at first. So I would recommend you look at growth, right? And so one of the things about juvenile toxicology studies is encourage you to make that call on growth and definitely use body weight and the long bone length, right? And the difference here is like, well, look at absolutes and then look at relative. And typically in adult, if it's relative, you know, you had an effect on absolute, but not relative, you'd say, oh, well, it's, you know, it's not test article related. But here, their bones are shorter and they're smaller weights, lower weights, then yes, you've affected growth, right? And you need to understand that and then go look at the other things when you're interpreting it, right? And those things, you know, can have effect on other organ weights, delayed sexual maturity, you know, and if there's any effects on bones, that's a little bit different than just effect on the length of the bone, right? So think of the skeletal system as its own target, different than looking at just growth. Right, and then also looking at the developmental hallmarks of maturity, right? If you know you affect growth or not, you need to understand that, and also, you know, pay attention to the timing of developing mammary glands too. That seems to be something that's not always thought about. And then we want to look at the big picture and see, like, do you have any effects, you know, that are different for adult findings, right? Or are they more sensitive? Just put a note here that the juvenile animals in the pre-weaning, they're not the same. The clinical, you know, observations that are made should be specific to juvenile animals, and you have to have your vets up to snuff on that, right? And when you're looking at organ weights, know that it's a dynamic model, right? And it's not going to be completely synchronous with how those organs grow to the body weight, right? You have isometric growth happening and then allometric growth. And please keep that in mind when you're looking at that data, and don't think of it as just like a stagnant model, kind of, so to speak, like how the adults are. And the background incidence is going to be different too, because you're not screening these animals as they would have been, like getting them to near adulthood and screening them for healthfulness. That doesn't happen on juvenile tox studies. So the historical background rates are different, and the reference ranges don't necessarily apply.
Yeah, so obviously we could keep going with so many questions, but in the interest of time, we are going to wrap here so we can have a little bit of time for audience questions, which everyone should be thinking about any questions they might want to ask and submitting it for us. But this is a chance for Susan to make a few last key points before we turn it over for those questions.
Okay, thanks, Christina. So what I want to just say is, like, what, you know, you're doing all these studies, hopefully they add value, and what does it mean for pediatric patients, right? So if you've identified, you know, a new safety hazard, you know, are there any effects on growth or development, or something that they should be looking for in the clinical trial, right? You're going to need to explain that, and that doesn't happen in the study report, but it certainly will happen in your regulatory dossiers. You need to be addressing each of these and describing the changes. Be very aware, like I mentioned already, like usually when these studies are conducted, you have large scale phase 3 trials going on. And so you need to be thinking about expedited reporting and communication between typically the CRO and the sponsor. It needs to be very seamless about that. Most of your pediatricians are going to ask, like, if you had a finding, is it reversible? Have you described that well enough? Do we understand it? Can we monitor for it, right? What is the margin of safety? Those are all key questions you should be able to be thinking about and answering. And then also know that this type of data typically does go in the label a little bit more than general toxicology data because it's specific to the pediatric patients. You know, there's specific sections in the label, and sometimes you're actually excluding certain age groups just based on non-clinical findings. There's quite a few examples that they're starting to develop from that. Okay, and then this is just my last slide, really summarizing it. And, you know, just saying, talk to the health authorities earlier and often. That's going to help you with just getting an efficient plan going, right? And it's going to have to be customized to the project. You're going to have to really think about what you need, and it's all that's going to be part of your PIPs and your PSPs, right? And when you write your study objectives, think about growth and development, right? And toxicity. All three need to be considered, right? And then when you're designing it and when you're going after it, I can't say enough, look at the comparative organ development. Right. And then you do have to dose range it. If I can, you didn't share all the examples, but there's so many I've learned, like really systemic exposure can really be quite unpredictable. So dose ranging and getting TK early is really going to be beneficial to you. Right. And so just to end that, you know, there's quite a few things that are specific to juvenile toxicology, right? And generally, like just as they say for pediatric medicine, the same applies here, right? As children are not little adults, juvenile tox is not little general tox. It's really a hybrid between your developmental and your general toxicologists. So that's all I had. And by saying thank you, Christina. And
Thank you so much.
I'm sorry, go ahead, Suzanne.
Christina, Susan, wonderful job. We've got a ton of questions from the audience, which is fantastic, and we'll answer as many as the time allows. So someone wants to know, in case you only have dog as a relevant species for gene therapy and you have to treat children less than 8 years old, what age dog should you select, assuming that rat and rabbit are not relevant species?
Yeah, so dog, I'm going to say it depends again, but usually it's going to be like a post-weaning, a few weeks old dog, I would say, several weeks old dog, 2.5 months or so plus. But really look at why it has to be dog, and if there's specific organ systems and the timing of the development of the dog versus an 8-year-old child, I think was mentioned. You get to make your argument in that sense, but I think it's going to be, you know, a pretty well past a post-weaning aged dog. Something like that is about as specific as I could get for that one.
How do you account for differences in GI physiology, for example, differences in fluid volume and composition between adult and other species and juvenile children?
Yeah, that's a good one too. So, I mean, again, right, it gets to like each of these, you're going to have to use that as your primary factor when you're talking about the different animals and the species choice, right? So I can't precisely say when, you know, what right now, which one for you, but certainly you have to be thinking about the difference between, and this is where it's a little bit different, and we have pre-weaning where they have like a milk-based diet and the pH is different, and the fluid retention, all those things are a little bit different. All I can say is you really have to do your research on the species of your interest and know why, and they'll be out there. And S11 has a lot of guidance on that too. And there's a good list of references that you can go to, which I'm happy to share too. I don't know if I put that at the end of this, but I'm happy to share those.
Yeah, they're at the end, and we can share those. What changes need to be considered for juvenile tox when targeting a pediatric orphan disease?
So those are discussions you need to have early on in your pediatric investigative plans, right? So it depends which organ disease and whatever, you know, so how you're going to develop clinically, I think you need to have that discussion for orphan indications. But it wouldn't change like all the scientific rationale for what you're going to do. That's not going to change. Those are scientifically based on the indication and the target pharmacology and such.
Great. This may need clarification. In ATMP and gene therapy, juvenile studies are single dose only. How long should the follow-up be at minimum? 1 month, 3 months? What do you think?
Yeah, so those ones are not covered by S11, most of them, right? And it depends on the species, and it's going to depend on if you have any specific concerns that you want to make sure you can evaluate, and how long you have to wait for them to develop those systems you want to measure. So I feel like I'm giving out I can't say exactly how long, but again, you can kind of see the theme where you have to rationalize it, on what your concerns were.
Makes sense. What is the youngest age that you've started dosing juvenile animals?
So I'll say for the rodents, and that's postnatal day 1. We did do a study where we dosed rodents on postnatal day 1, but that was to support 3rd trimester women, right? We were doing something specific for preterm, that we were intending to give to pregnant women. So yes, it was a juvenile toxicology study, but that's what you needed when you did the comparison to the human. So I don't think if you're doing a pediatric population, there's no reason to go that young in the rodent.
So Susan, can I follow up on that? So is that oral dosing? How did you do that?
Yeah. Oral gavage was very doable. And so in that case, and again, the team that did that was the reproductive toxicology team, technical team, right? So they're used to working with fetuses and really young animals and littering very young animals for pre and postnatal studies. That's where you really have to have that team who can do those things, right? Because the hardest day of dosing is the first day when they're the youngest. So you don't get any like warm up to it. So yeah, they're technically a challenge. You just have to really strongly rationalize why you would do it, right? Because you're going to compromise being able to do the older ages, and it's just technically really, really difficult. So, I would say postnatal day 4 at the very youngest for most pediatrics, and that's when you're going down to preterm infants, right? So usually postnatal day 9 is a good age to start rodents.
Someone wants to know whether you recommend sharing your juvenile tox protocol DRF data with regulatory agencies so they can weigh in on your dose selection and study design.
Yeah, I have not gotten dose selection feedback from the health authorities. I typically don't find that that's a place of strength for them. Unlike in the CARC, right, when you're doing the whole CAC response and the carcinogenicity, they're very, very clear and very into, agreeing on the dose selection. But in the juvenile toxicology space, I've not done that when it comes to dose selection. If you had unexpected findings or things you're difficult to navigate, probably it's always good to have that dialogue. But usually there's not enough time for a lot of dialogue once you've done that. My recommendation is you get agreement just on the overall plan before you initiate the dose ranging study.
Looks like we just have 2 more questions. How do you recommend adjusting for age between human and animal species?
Yeah, so there's that figure that I shared that's pretty complex, and you're going to have to give some trade-offs, but that's where you have to go to, like, where the organ systems are in development, the comparative organ system. If you have concerns about the lung, then you have to go look in that species and really do that. So there are some things out there that kind of oversimplify that, and I don't think that that's actually doing you a favor, because it really needs to be aligned to what your concerns were for that project.
Looks like we have one last question. Someone says toxicokinetic assessments may be challenging due to small sampling volumes in juvenile animals. Is sparse sampling an acceptable alternative?
Yeah, I love that question. Thank you for asking it, because it's not something I touched on, and it is a key message. I should have touched on it. Yes, sparse sampling, totally fine. And a lot of times you're just trying to get an assessment of where you are. 3 time points, and we say we do like modified AUCs. They're not exactly AUCs, and your DMPK colleagues may not love you turning it into an AUC data, but it's totally a thing you can do, right? Because you're talking about taking animals for each time point. And the other part is you can try to really push and get a micro sampling technique going and using those, because it really matters how many animals you use, but you need some TK information. Even if it's ballpark, it will be helpful for you.
Wonderful. Thank you so much, Susan and Christina. I think we've learned so much about best practices in juvenile toxicology from you. And thank you so much to the audience for all your attention and wonderful questions. Before concluding the webinar, we just have a few announcements. We just want to let you know that we've got a ton of great webinars coming up for the rest of the year, and you can sign up for all of them by visiting us at certara.com. On behalf of Certara, I'd like to thank you for attending today's presentation. This concludes the webinar. Goodbye and have a great day.
Bye. Thank you.
Bye.